One of the biggest challenges in nutrition science is figuring out why people have such different metabolic responses to the same food. It's clear that our own genetics can't explain all of this variability. So, we're going to examine the role of the microbial metagenome. That's the collective genome of all our gut microbes as a critical link between what we eat and our metabolic health. We'll focus particularly on cardiometabolic disease and the move towards personalized nutrition. Here's how we'll break it down. First, we'll find the problem of metabolic variability. Then we'll talk about how the microbiome develops, how diet acts as a selective pressure, and how microbial metabolites work as signaling molecules. After that, we'll look at atherosclerosis as a specific case study of this diet microbe host relationship, and we'll finish up by looking at where this is all heading with precision nutrition. All right, section one, let's talk about that microbial influence. We're positioning it here as the main hidden variable that really explains why our metabolic responses to food are so personal. So, this is the central question. Why is there such a huge difference in how people's blood sugar, fat levels, and inflammation markers change after eating the exact same meal? The investigation into this puzzle keeps pointing to one critical factor: the gut microbiome. To understand the power of our gut microbiota, just look at the genetic potential. Our human genome has around 23,000 genes. In contrast, the microbial metagenome, the collective genome of our gut bacteria, has over a million unique genes. This represents an enormous toolkit of metabolic machinery for breaking down parts of our diet that our own bodies simply can't digest. Now, how does this complex community get there in the first place? Let's look at the development of the microbiome from the moment of birth all the way to a stable adult community. The process starts at birth, and right away, the mode of delivery makes a big difference. Vaginally delivered infants get their first microbes from the mother's vaginal and fecal microbiota. C-section infants, on the other hand, are mainly colonized by microbes from the skin. Those first 1,000 days are a really critical window. The community is simple and changes a lot. But by about age 3, it matures into a much more stable and complex system, looking a lot like in adults. So, what does that adult microbiome look like? Well, for one, it's pretty stable over the long term. Most healthy people have a community dominated by a few major groups like Bacteroideus and Firmicutes. But here's the key part. When you get down to the species and strain level, there is a huge amount of variation from person to person. And it's this variability that helps explain those different responses to diet. This brings us to the single biggest factor that shapes our adult microbiome: diet. You can think of it this way: the food components that make it to your colon act as a powerful selective force. They encourage the growth of microbes that have the right tools to break them down. Let's take fiber as an example. Its availability has a direct impact on the health of your gut barrier. A diet high in fiber feeds the fiber fermenting bacteria, which helps keep the whole ecosystem diverse and keeps the protective mucus layer in your colon intact. But what happens if you don't eat enough fiber? Well, some of those bacteria are forced to find another food source and may start to consume the mucus layer itself. This erosion of the barrier can increase gut permeability and is thought to promote inflammation. It's not just fiber either. The type of protein you eat matters too. Animal-based proteins tend to select for microbes that can tolerate bile and provide the raw materials for some potentially harmful metabolites. Plant-based proteins, on the other hand, can be used by microbes to produce beneficial compounds. And beyond just macros, we have to consider specific things in modern diets that can mess with this system. Common food additives like emulsifiers can alter the microbiota. Some artificial sweeteners have been shown to impair our glucose control through microbial pathways. And we now know that high saturated fat intake can contribute to insulin resistance, but it does so in a way that depends on the gut microbiome. So, how do these microbes actually influence our body's physiology? The main way is to the production of small molecules called metabolites. You can think of these as the chemical signals that allow for communication between the microbial world in our gut and our own host systems. A classic example of this is short chain fatty acids, or SDFAs. These are the main byproducts when microbes ferment dietary fiber. One of them, butyrate, is the primary fuel source for the cells lining your colon, but it's also a signaling molecule that helps regulate immune responses and can even trigger the release of hormones that make you feel full. Okay, let's pull all these ideas together and walk through a really important clinical example: the connection between diet, the microbiome, and the development of atherosclerosis, or hardening of the arteries. The key molecule to know in the story is trimethylamine N oxide, or TMAO. This is a metabolite that promotes atherosclerosis. We know that high levels are linked to a greater risk of atherosclerosis and blood clots, and its production is a great example of a team effort between gut microbes and our own bodies. Here's how the pathway works. First, you eat foods rich in precursors like L-carnitine and choline, think red meat. Second, specific gut microbes take those precursors and metabolize them into a gas called trimethylamine, or TMA. Third, that TMA gets absorbed, travels to the liver where your own enzymes convert it into TMAO. And finally, that TMAO circulates through your blood and contributes to the formation of plaque in your arteries. Understanding pathways like this one is what allows us to move beyond one-size-fits-all dietary advice and toward what we call precision nutrition, where we can tailor what you eat to your specific individual microbiome. And this isn't just a theory, we see it in studies. For instance, in one study where people were put on a whole grain diet, the individuals who started with a high abundance of a bacterium called Prevotella lost significantly more weight than those who had low levels. This shows that the makeup of your microbiome before you start a diet can predict how well you'll respond. This opens the door for some really interesting new interventions. We're talking about next generation probiotics designed for specific tasks, like helping to regulate appetite. There's also the concept of postbiotics, where you just administer the beneficial metabolites directly. And finally, precision prebiotics, designing specific fibers to feed specific beneficial microbes you want to encourage. So to wrap things up, let's quickly review the core principles we've covered about how diet and the microbiome interact. Here are the key takeaways. 1, the microbial metagenome gives us a huge boost in metabolic capability. 2, diet is the main driver shaping the structure of that microbial community. 3, microbial metabolites are the key chemical messengers in the conversation between our microbes and our bodies. And 4, the microbes you have to begin with are a critical factor in determining how you'll respond to any dietary change. To make sure these concepts has landed, take a moment to consider these questions. First, can you explain the mechanism by which not eating enough fiber might lead to inflammation in the gut? Second, can you trace the metabolic pathway from eating L-carnitine all the way to atherosclerosis, making sure to note the roles of both microbes and host enzymes? And third, in the context of precision nutrition, what exactly do we mean by a responder? What do you think the answers are? Leave your answers in the comments. And if you like this video, hit that like button. And please consider subscribing to my channel.